Right, good morning. Welcome to Science Seminars. Okay. I'm going to turn off your Wi-Fi on all your Wi-Fi devices to help us enable our recordings here. Okay. Welcome to our new home in B103. What do you think? Yeah?
Temporary, right?
Temporary. I know it's harder to sleep in this room because it's more well-lit and stuff like that. But we do have better tables, so you can get more homework done that way, which you wouldn't be doing during seminar class. All right, today we have for us Dr. Michele R. Harris, okay? And she has a degree from the University of Arkansas in biochemistry, a PhD in biochemistry. She has her bachelor's degree from the University of Central Oklahoma, okay? And that's also in what, chemistry?
Yes.
I think. Okay. And she's been at Austin since 1994.
Yes.
So she's been there for a long time. She started a biochemistry program over there, I think. And she's got wonderful things to tell us about biotransformations. Okay? So if you want to know how you can go from a caterpillar to a butterfly, this is the place to do it. Also want to remind you that next week is spring break. So we will not be meeting for seminar next week. Yeah, I know you're all broke up about that. When you come back from spring break, the week after spring break, we have a very big event on our calendar with respect to our science seminars. Gym Tour from Rice University will be up to give 2 talks. One on Wednesday night on being a Christian in the sciences and his own personal story. And then Thursday morning at the regular time, he'll be giving a science seminar on nanocars and nano people and things like that. Jim Tour is one of the 10 most cited chemists in the world, right? So he's a really big name in the chemistry world. So if you want to come to a really big event here in the science seminars, you want to come for that after spring break. Right, so with that introduction, let's give Dr. Harris a warm applause.
Thank you. I'm glad I came before Dr. Tour came instead of after. Wow. So, as you said, my name is Michele Harris. I've been at SFA since 1994. I went there straight after I finished my PhD in biochemistry. Wear a few hats there. I'm the assistant chair and graduate advisor. And 5 or 6 years ago, we started requiring 2 semesters of research in our chemistry degree as a part of the curriculum. And so all the work I'm going to present today has been done by undergraduates under my supervision. And so it's been a good transformational experience for our department, for our students. It's benefited them. And I'll show you some stuff that undergraduates are able to do as a part of their curriculum now. So the title of my talk is Biotransformation Reactions, Conditions, and Antimicrobial Properties. So what are biotransformations? Well, yes, caterpillar to butterflies are biotransformations. That's not exactly what we're thinking about in a biochemistry setup. So a formal definition, a chemical conversion of a substance into a desired product with some sort of whole cell or living organism. Okay? So what happens is these living organisms or whole cells contain all the enzymes necessary to carry out chemistry. We do it all the time. We metabolize food. There's all kinds of things. If you think about, have a picture of grapes, you could even talk about biotransformation as photosynthesis and plant and all of that and get into the biology aspect. Or you could think about how about 3,000 years ago, Egyptians and some other cultures began to learn to make wine. They used a yeast. Yeast has all the necessary enzymes to make wine. So that's kind of a broad overview of how you can look at a biotransformation reaction. So what are some applications of biotransformation in general before we get to ours? A lot of pharmaceutical applications. We'll talk more about that in a few minutes. More recently, if you think about the oil spills that we've had, microbial transformations of pollutants to less hazardous substance, things that will degrade oil to less hazardous substances, coming up with ways to modify those genomes. That gets into a little more biochemistry aspect of it. That's been done. And so here's a picture from the Gulf after the big BP accident. People are down trying to clean that up. What other kinds of things? We won't get to it today. Cosmetics, a huge multi-billion-dollar industry. Scents, flavorings, all of that falls under. Can we get an organism to do a particular reaction efficiently? Yes. Food processing, making cheese, all kinds of things use enzymes for that. Agrochemicals, growing pesticides, insecticides. Can we make something and can we get an organism to do it in an efficient manner? So why did we start using biotransformation? So in the American Chemical Society, we really are starting to think about green chemistry. There's a whole branch of that, a lot of publications from ACS about it. So if we can reduce the need for harsh chemicals, or for SFA, Department of Chemistry and Biochemistry, for us to not have to pay for disposing of chemicals, we like that locally as well on our budget. So yes, there are ways that we can have more mild reaction conditions using biotransformations. We reduce the need for harsh chemicals. It's more of a green chemistry as far as the topic goes. And for the particular reactions we're looking at, we have an easy recovery of the product. One other aspect that's important to us, and if you've not had organic, I want to talk about this a little bit more, is stereoselectivity. So a lot of enzymes are either stereospecific or stereoselective, and I want to talk about those words before I get into my research project, because I wasn't sure here that everybody would have had organic chemistry. So stereoisomers are a broad class of isomers. They're compounds that have the same molecular formula, same number of carbons, hydrogens, oxygens, nitrogens, whatever's there, but they are arranged differently somehow in a three-dimensional space. In organic chemistry, most often that comes up with cis-trans isomers, so if you've had organic, you've heard of those before. One of the next topics in organic then ends up being enantiomers, which is the next word that I have up there. And enantiomers are non-superimposable mirror image molecules. The easiest way to think about enantiomers are your hands. So my hands are a mirror image of each other. If I have a mirror between them, if I try to superimpose, I can superimpose my fingers, but I can't superimpose my thumbs. So all living organisms and enzymes, almost all, recognize one of those enantiomers from the other. So it's this whole area within organic chemistry that bleeds over into my specialty, biochem. So I drew a little molecule here. It's got some central atom. And it's got 4 different groups, and I just made different colors, black, blue, and white. This plane here would represent a mirror between the two. You can imagine if I pick up this molecule on the left and try to superimpose it or set it on top of the one on the right, the red and the blue would line up. They would superimpose. But the white functional group would be on top of the black atom, and the black would be on top of the white. So they're non-superimposable mirror images. Our bodies can make, and organisms can make one of those enantiomer over the other. Occasionally they'll make mixtures of both. That's called a racemic mix. But biotransformations give us a good deal of stereoselectivity where it would make one of these particular type of enantiomers in high excess over the other. Stereospecific would mean it makes only one of them. Stereoselective means, oh, it makes 95% of this, maybe 5% of the other. All right, any questions? Anybody not in organic that would like to hear more about enantiomers? Okay. So a little bit more about them. They have the same molecular formula and they have the same kind of physical properties, melting point, boiling point, solubility. So some common methods of separation don't work to separate enantiomers. They do differ in how they rotate plane polarized light, and they do differ in how they react, especially in biological reactions. So I picked an example. Ibuprofen. Everybody here, I'm sure, has taken an ibuprofen at some time. I pulled the structures from the web. So I have R-ibuprofen listed here, and I have S-ibuprofen here from a website. So R-ibuprofen, and just a little bit more of the organic if you've not had it, that dash means it's coming out of the plane of the screen, and the wedges, as we call them here, mean they're going behind the plane of the screen. So they differ at that one carbon there, and that is a chiral carbon because it has 4 different groups attached to it, and a carbon with 4 different groups will rotate this plane polarized light differently, and they react differently. I put an asterisk here by the ibuprofen because S-ibuprofen, the way we name the enantiomers, we don't want to get into that today, but the 2 enantiomers, one's S, one's R when you look at an enantiomeric pair. The S-ibuprofen, the figure on the right, it's the one that's physiologically active. it will inhibit what's called a COX inhibitor that causes inflammation and pain. So you know you take ibuprofen, you've sprained your ankle's swollen, you take ibuprofen for the pain, you also take it for its anti-inflammatory effects. So the S-ibuprofen binds because of its three-dimensional arrangement in that enzyme and it inhibits it. The R does not. So they can sell what we call a racemic mixture because this one is not harmful at all. It doesn't really have any effect. This one has the physiological effect. So if you've had organic before, I'm sure you've heard of the thalidomide famous example, the first kind of set of enantiomers that cause problems. Thalidomide was a great drug that stopped nausea. So pregnant women in their first trimester, nausea, right? That's a common side effect. So in the early 1950s, they gave it to pregnant women to stop nausea, and it ended up causing severe birth defects. As they studied that, one isomer had more of the anti-nausea effects, the other enantiomer caused the birth defects. So now, every drug that's produced, we take into account if there are a possibility of enantiomers. It has to be studied individually, each isomer, each enantiomer, before it can go to market. All right, so enantiomers. We are interested in that concept because biotransformations tend to make one kind of isomer over the other, and we've studied some with my undergraduate students at SFA. We used Daucus carota, carrots, and here's some things about carrots and other vegetables that we have investigated. So we know that whole cell biocatalysts have been a lot of advantages for those. If you think of vegetables especially, they're widely available and inexpensive. I can go to Kroger all year long, buy carrots. We could get the biology department to grow them up. They already have a method of recycling any other coenzyme. I'm going to talk about this NADH in a minute. Most biological reactions, not only does the enzyme catalyze the reaction, they have an organic molecule that assists in that in some way, so hence the name coenzyme. And again, I've already mentioned the stereoselectivity. So what we began doing when we were looking at this, if you have some prochiral ketone, so a ketone is anything that has carbon groups, a carbon with a double bond oxygen on it as a ketone. So if you take this prochiral ketone and you put some plant tissue, like our carrot strips, what you have the option of making is reducing that ketone to an alcohol, and the cofactors and the enzymes can be recycled to continue catalyzing that reaction in a living system. So again, I'm showing you the three-dimensional result of that. If the OH group adds behind the plane of that, that's a trigonal planar shape, then the OH will be behind, and the dashed lines represent going behind. If the reaction occurs by the reactants coming together on this side, then it would come out of the plane of the screen, and that's why you would have the wedge there. So people have been looking at those. Chiral means handedness, and it's a molecule that has 4 different groups attached to it. Prochiral means that once it goes into reaction, the product has the ability to be chiral, it can be stereospecific. So don't go cross-eyed, okay? I'm a biochemist. You have to show some kind of coenzyme structure before you go on for those kind of things. So I took this from the biochemistry text I'm using this semester. NADH is nicotinamide adenine dinucleotide, and it's reduced. It's a very common coenzyme. So this, oops, wrong button. This is the NAD. So you see why biochemists want to have some abbreviation. Nobody wants to draw that. All right, it's got an adenine ring like you see in DNA. It's got ribose, a diphosphate linkage, another ribose, and then this pink portion comes from niacin, a water-soluble vitamin. We hook all that together, and it's a coenzyme in our bodies for that. So if we ingest ethyl alcohol, we have an enzyme called alcohol dehydrogenase that will oxidize that ethanol to acetaldehyde. Okay? So there's an enzyme that does that, but the coenzyme in this reaction, and many like this in metabolism, is this NAD. You can't have oxidation without reduction, freshman chemistry, right? If you have one, something else has to be reduced. So we have the oxidized form. It's got a formal charge there that's got a positive on it. During this reaction, the coenzyme is reduced. So we've messed up some of the conjugation of the ring a little bit, and we've added hydrogen here. And we have the reduced form NADH. You see why biochemists use these abbreviations. It really has a positive charge and it really picks up a hydrogen to simplify what's going on. So as the ethanol is oxidized to acetaldehyde, the coenzyme is reduced to NADH. Then there's some method to regenerate NAD and continue in the body, in a living cell. So every time we say NADH, it's this big old structure here, okay? All right, so let's get back to kind of the research project that my undergraduates and I have been working on. So chiral alcohols have a lot of applications to pharmaceuticals. Again, lots of different people have looked at, wow, these whole cells are really phenomenal at making one isomer. We are designed to be that way. The enzymes are that way. So how can we apply these in the lab, perhaps? Then people began not only looking at things like baker's yeast and bacteria, because if you do something with baker's yeast or bacteria, when you lyse open those cells, everything that was inside the cell, plus the one thing you're after, you're finding a needle in a haystack. So people began looking at what other kind of whole cells. What if we use these vegetable strips or plant tissue from apple, carrot, sweet potato, onion? And they started finding out that, hey, these things can also do stereospecific reactions as well. I'm not looking for a needle in a haystack for purification of the one compound that I'm interested in after that. So people started looking in the early 2000s at different ones. They found that good old-fashioned carrot and potato, pretty inexpensive, tend to give pretty good yields, 50% or more. That's not a bad yield. And they tend to have purest products in that they're pretty enantioselective, where they can get one enantiomer over the other. So acetophenone, if you've not seen it before, is this structure. It's got a benzene ring, and it's got a ketone. So this is my prochiral carbon there. And that was kind of the first aromatic compound that some of these groups started working with. And what they found was often the carrot produced the S isomer in high percentage yield compared to the R isomer. And they found that the potato tended to produce the R, but not as quite a high purity. How cool is that? I mean, it's like potatoes, you know? Carrots. So we looked at the literature and, again, had some undergraduates doing this work. So we found a reaction that someone else had said, Rabia, had said that made the S isomer of this particular molecule, and we wanted to study that reaction. So benzofuranomethyl ketone, BMK, was the abbreviation we came up with in our lab. And then in the presence of carrot fragments, that's it, and water, we get a S isomer of the alcohol out, and we can purify it. Okay? So somebody else had reported that. We wanted to explore that reaction and try to understand how and why that's occurring. So why would a benzofuranomethyl ketone, kind of a mouthful, do this? So we kind of started looking at some natural product literature and found that a lot of plants naturally produce benzofuran derivatives with all kinds of functional groups off of them. And some of those different compounds have been cytotoxic to other cells. They have antimicrobial properties. They're killing bacteria. They are anti-inflammatory properties, all kinds of things. You can look through natural products, the Journal of Natural Products, find thousands of examples of benzofuran derivatives. So again, a benzofuran ring is a 6-member benzene ring with a 5-member furan ring. And one example of that, I'm not even going to try to pronounce the name of that compound. But anyway, this particular plant here, Algaea oligophylla, when they took the twigs, ground them up, and started isolating all the organic compounds out of them, they found this lovely benzofuran derivative. Here's my benzofuran, and it's got a boatload of other kinds of things around it. This was a very strong insecticidal agent. It grows in Malaysia, Singapore, Southeast Asia, some of those kind of places. Again, you can find hundreds and hundreds of examples of some kind of benzofuran that has some sort of physiological property. So we wondered, well, do carrots make this? Or, you know, because we're adding it artificially in our lab. Would they have some sort of similar reaction, and if why would they? So we began exploring this reaction further. So I had several students that began working on this about 4 years ago. So we wanted to figure out how we could isolate the pure BMA, the alcohol, stereospecific alcohol that this carrot would make from the benzo methyl ketone. So again, BMA and BMK are much easier to say when you're talking around the lab than the whole long name every time. So we figured out, over about a year, you take 8 grams of carrot strips, wash the carrots, peel them, and we grate them with an actual kitchen peeler. And we tried every part. We tried the epidermis, we tried the cortex, all of that. We had some of that. But if we took the outer portion, take off the epidermis, 8 grams of carrot strips, 25 milligrams of the ketone, the prochiral ketone, BMK, that's about 1.6 times 10 to the minus 4th moles, and 25 milliliters of deionized water, of all things. Set it up in these falcon tubes. Okay, so each one of these, we weighed 8 grams of carrot strips, 25 milliliters of water, 25 milliliters of water, 25 milligrams of our starting material. We put on the rotisserie. One poor student had the whole assignment for almost a whole semester. How long do we let it mix? How many grams? What's the good ratio? So she spent the whole semester helping us figure out this works well. So in 2 hours, the reaction will go to completion. So this is a great project for undergraduates who come and go. You can come in, do your stuff, get it all set up. You can come back 2 hours later and do the next step. So it really has been a nice project for undergraduates to work on from that standpoint. We then do good old-fashioned vacuum filtration. We extract 3 times with equal volumes of ethyl acetate. We take it to the rotovap and remove the ethyl acetate, and we're left with a crude product. We then pour a silica gel column, and because carrots have some lovely carotenes and other things that we have to get rid of, they stick to the column longer than our alcohol does. We use thin layer chromatography, TLC, to verify the product, and polarimetry. We also did some IR originally to make sure we had what we had and some NMR. I won't have any of that data with me today. So a TLC plate is if you've never seen one, is a glass plate, and it's coated with white silica on it. Ours has embedded in it a UV indicator, and any place an organic molecule will show up as a dark spot on it, and then we take a picture of it. So what we did for the TLC to verify our product is we purchased the BMK from Sigma Chemical Company. We make up a standard. We use 2 microliter automatic pipetter. I'm a biochemist. We don't use capillary tubes, and we put exactly 2 microliters on of our standard. And then out of the extraction, we take 2 microliters of sample, and you can see these spots don't travel as far. That's the alcohol. The solvent we're using is an 85-15 hexane ethyl acetate, a very nonpolar solvent. So the BMK is more nonpolar, so it travels further. The OH group has a small amount of polarity, it doesn't travel as far. So based on the physical properties of those. And I marked on top of them, or I wrote on top of them, but there's no other spots here. So we have a single product here for our alcohol that we produce. After right before we do silica gel, we took a picture one day. This is our orange oil that we got from the carrots that still contains a little bit of the carotene from the beta-carotene from the carrot and our product. So, water's the best solvent. And I can tell you that today because we tried a boatload of other solvents. We tried buffers. I'm a biochemist. Well, maybe the enzyme would be happier and be more active if we find a good buffer. Maybe pH matters. Maybe temperature matters. So we decided we wanted to see the best reaction conditions for this conversion. All the biotransformation literature we were reading was coming from organic kind of labs, and they just were like, hey, it pops out and we're analyzing it with NMR, so we were trying to figure out the reaction some more. So we use various buffers, pH, temperatures, and detergents, and I won't give you the whole list. So this was another student's whole semester doing the same thing under all these different conditions. And the result was, really, water at room temperature was the best. So the whole semester told us that. So I picked a few. I didn't pick all of them. So we did it at 4 degrees Celsius refrigerator temperature, room temperature, and we did it at 37 degrees Celsius. And there was no big difference in any of the yields, more than 1%, and that's not even enough to tell. So we stuck to room temperature because it's a lot easier than trying to get in and out of the incubator or in and out of the cold box. So room temperature. And again, I just picked a few. Acetate because it was pH 4.5. 1X TBS is a Girl Biochem buffer. Tris buffer with saline. It's a common buffer at pH 7.4. And then at the other end, to show you some representative data, we tried 50 millimolar carbonate. We did some phosphates. We did different Tris buffers. We did quite a few others. And then we did old-fashioned water, deionized water for that. We calculated our percent yields. We didn't get anything in the acid environment. At 7.4 in TDS, we got a 33% yield. The specific rotation of the product was -15. The literature value for our alcohol should be a -16.4 to indicate that it's a pure benzoyl-methyl ethanol for that. At 50 millimolar carbonate, we got a 29% yield, and we were a little bit off there on what the optical activity was. And in pure water, we got a 57% yield, and we were within 0.4, which is about the limit of detection of our polarimeter that's about 15 years old. We ran some standards with sucrose, and we usually were getting between 0.3 and 0.4 of the accepted literature value. So we felt like that was good, and that's what we used for the rest of our studies. So all of that to find out, hey, water was the best to start off with, and we didn't need the buffer. So then I had another student, and we were trying to figure out, is there a way to get this enzyme off the surface of the carrot? Can we purify them in a biochemistry? After all, we need to purify some protein and study it. And so we had one student that was trying to see if, under these various conditions, any of that protein was coming off and active just in the liquid over there. So we take the carrots out, just filter it, and see it. And it was completely erratic result. Yes, it made BMA. No, it didn't make BMA. And we're like, what is going on? So one student and I sat down and we're like, okay, so what is the properties of that targeted protein, and what can we do to figure out why we're getting such inconsistency? So what we did was we noticed if you mix carrots overnight, there's a lot of tiny particles of carrots. Even though we have strips, sometimes it breaks off and sometimes it's really small particles. So after this inconsistency that we observed, what we did next was we set up our setup like I talked about earlier. We had a falcon tube, we had 8 grams of carrots, we didn't put BMK in it, and we mixed it for 2 hours. So in one test tube, what we did was we looked at the supernatant. So we visually just poured that off and kept the big pieces of carrots from coming with like a spatula or something. But when you held that up, you could see some particles still from the carrot in it. Then we took another sample and we vacuum filtered it twice. And we kept the filtrate, and then everything on the second piece of the filter paper, any particulate matter that was there, we saved. We let the filter paper dry. So then what we did was we put that in a petri dish, and you can see the orange off our second piece of filter paper. You see some little white crystals, even a piece of carrot there floated off. And we added some water back to it, and we added some BMK to it, and we mixed it very gently on top of an orbital shaker. And what we got was, with just the supernatant, where we physically poured it off so we knew we had carrot particulate, we saw the alcohol being made. In the carrot filtrate, where we vacuum filtered the substance twice and just kept it, there was no conversion. And on top of the filter paper, those few pieces or particles of carrot that were left were enough to show some conversion to alcohol. So our inconsistency had been our own fault. We weren't really treating the samples all the same. So we figured out from that, we aren't pulling, at this point, any of the active protein off the surface of the carrot. It's staying anchored somehow on the surface of the carrot. Our BMK must be interacting with it in one particular orientation so that we're getting one isomer only. So then we started thinking about some other things. How many times can we use the carrot? The whole idea is green chemistry. We just have carrots. We can compost those. And we were using them once and throwing them to compost. So how many times can we use them? How long, since we plucked the carrot out of the ground, can it recycle the cofactor? Because after we pulled it up, it's really not alive anymore, right? We're going to eat it, or we're using it in the lab. So how many times can a single set of carrots be used before we lose activity of this by the coenzyme or the enzyme? Is there a problem with that? So what we did, another student worked on this one. Same kind of setup. You grate the carrots, you put the water in, you put the ketone in, you let it mix for 2 hours. Then after 2 hours, we did a vacuum filtration. We kept the carrots. We rinsed the carrots with deionized water. We put them in a fresh tube, added 25 more milliliters of water, 25 milligrams of starting material. 2 more hours, it was a long day. And then we did it again, and we did it again. We did it 5 times. So what happened when we got that? So here we didn't take our picture of our TLC plates under UV light. I don't know why a student didn't, but we drew circles with them so we could look at them. So what we have from left to right is this first one is our first use. So here's our standard BMK that we purchased from Sigma, and this is our alcohol spot. There's no other spot here that the students circle. So then in the second use, we still have complete conversion of the ketone because we only have a single spot showing the alcohol. But if you notice in the third use, we still have a ketone standard, but here now this spot was getting fainter, and now we suddenly have a second spot. We had some ketone left in our sample, so we hadn't converted all the ketone to our alcohol. Fourth time, the ketone spot got darker, this got lighter. Fifth time, you could barely see the alcohol spot, and it was predominantly ketone based on just the intensity of the spot. So from that, we realized with our setup, we can use the carrots twice and get complete conversion. So we'd have less to throw out even as far as carrots, or we could use a little bit less from that. So again, back to the question, why does a carrot or a potato do that? Okay, they grow on the ground. Is it some protective method? Does it have some natural reaction that's similar to this one that we're forcing it to do? So a different student who was a biology minor and I talked to him, well, let's see what we can figure out and why this reaction might occur. So we wanted to see how the ketone starting material or how our alcohol that we were producing affected the growth of bacteria. We started with 25 milliliters of LB broth, which is the standard culture if you've ever been in a micro lab. That's probably the smell of the broth. We inoculated it with 20 microliters of E. coli. That's a non-virulent strain that we just had available in the lab for cloning, BL21. We did our appropriate controls. We grew the bacteria in one flask. And then we decided to use DMSO as the delivery method. Dimethyl sulfoxide is the name for DMSO. It is very commonly used in these kind of studies to deliver a drug to a tissue culture because it doesn't have a large effect on the growth of the culture, of the bacteria or the cell culture. So we did our standard curve. The blue line is bacteria with no DMSO, and the red line over 9 hours was the growth with DMSO. It was a single run, so I don't have statistics on that difference, but there's a little bit of a difference. Based on that number, about 9% different. So that kind of matched with literature. 5–9% didn't seem out of order of having a little bit of effect of the growth of the bacteria. So then we took our setup, exact same kind of setup. We used the same bacteria we had in lab, the E. coli, BL21. We used DMSO, and we're using like 50 microliters is all of that to deliver it to the flask. And we dissolved either BMK in the DMSO or BMA in the DMSO, and we had 2 sets for those. We grew broth only and made sure we were following good sterile techniques and that it didn't start growing. We grew some with bacteria. We wanted to double check, and we just added the DMSO to see if there was any effect there. And then we had the broth, the bacteria, and DMSO. So we had all those controls. For growing bacteria, you measure the absorbance at 600 nanometers. If you're a biology person, that's called OD 600. Optical density is how biologists talk about it. Chemists talk about what wavelength we're at, and we're measuring. That's a standard place for measuring cell growth. Don't get lost in all of this, but this was our ketone, and we added it at approximately 5, 10, 20, 30, 40, and 50 milligrams to the various flasks. We grew bacteria with DMSO and no compound in it, and so that's the blue line. We added a little bit of our 5 milligrams nearly of the starting material, the ketone, or the 10 milligram. Not a big effect, and again, we did one trial of this, so I can't talk about statistics of that. That was just our observation from that. And then, as we started adding higher amounts up to 40 and 50, we continued to see a decrease in the absorbance, which means there was a decrease in the growth of the bacteria. So BMK was inhibiting the bacteria growth. We did the same thing with our alcohol product, and we saw the same kind of trend, except the effect was more pronounced, as 5 and 10 milligrams didn't affect it much, but 20, 30, 40, and 50 continued to inhibit the growth over time. That's a lot of lines to look at. So I went back and pulled out just the 50 milligram data, the 48.6 for that. So here's the bacteria by itself. This is with our ketone added at 50 milligrams. and this is with our alcohol. So a little bit more effect. Maybe just looking at the numbers here, that's a 17% difference between the alcohol and the ketone. So it looks like the alcohol is about 17% more inhibitory toward the bacteria. So maybe it does, maybe there's a similar reaction in carrots, and maybe it's providing some protective function. So then we wanted fungus grow on, right? Everybody's bought fruit with mold or what all on it. So we wanted to look at that. And I went over and talked to the biologist that's a fungus expert. And she's like, you don't want to do that first. I'm like, okay, I'll take your word for it. She said, start with baker's yeast. It's a simple eukaryote that's cheap and easy to handle. If you get a good handle on that, I'll help you with the real fungus later. Okay, so we did that. We used baker's yeast. We followed hers. Yeast have a longer replicative lifespan than bacteria, so we could take the measurement every 1.5 hours. We did a growth curve. We did the controls and all of that. I won't go through tons of more spectra or Excel graphs of our data from that. So this is the data that we had from the bacteria. And the alcohol was about 17.5% more inhibitory on bacterial growth than our ketone. What we saw in yeast, which are eukaryotes, is about a 35% difference. So twice the effect. So it seems to be more active against a eukaryote than a prokaryote. So all of those, we've done one set of data with those. Another student, I had several, I keep saying that. Well, what can we do about this protein? We weren't pulling it off in our earlier studies. Is there any way that we could go about trying to isolate the protein from the surface of the carrot? So I won't give you all the gory details. After lots of different trials, lots of different buffers, lots of different detergents, we settled on, for a variety of reasons, using the tris buffer with saline and 25 millimolar N-octoglucoside. That's a detergent that's known to be good for plants. It has a low critical micelle concentration, a whole bunch of other reasons. And so we couldn't do the gentle rotisserie, so we put 200 milliliters of the buffer, 20 grams of carrot strips in a big Erlenmeyer flask and put it in an incubator shaker. We left it at room temperature, and we shook it overnight at 250 RPM. So we agitated it quite a bit for that, hoping that it would begin to pull whatever enzyme catalyzes this out of the surface of the carrot. We figure it must be a membrane-bound protein to be in the surface like that, so we wanted that detergent to mimic the membrane. We vacuum filtered it, we centrifuged it because we had a lot of particles still left because we had been so much more aggressive with it. And then the resulting supernatant, about 200 milliliters, we concentrated in something called an Amicon concentrator. If you've never been in a biochem lab, an Amicon concentrator is a tube that fits in the centrifuge, and it's got basically dialyzing membranes anchored in it at an angle, and it has a semi-permeable membrane that allows molecules that are small through, and it retains large ones. You can purchase them with different molecular weight cutoffs. We had a 3,000 molecular weight cutoff. Most proteins are bigger than 3,000. Most small molecules would go through, and we would concentrate down the protein portion and let the other small molecules go through. So there was a day or two doing that, to say the least, and we wound up from 200 mils with about 1 mil in a microfuge tube in our biochem lab. We took an aliquot of that, and we realized after we let it sit, another precipitate still formed. So we centrifuged it. We could kind of see particles there. We centrifuged it in a microfuge for these small tubes, and we took the supernatant. We also tried to resuspend the pellet to see if there was protein there. We didn't have a lot of luck with that. But this supernatant, we ran an SDS page gel. SDS page gels separate proteins by size. It's a super common biochemical tool that we use all the time. And so you purchase standards from the company, you run your sample. We did it at a 40-fold, 100-fold, and 200-fold concentrate. And where the darker bands are, or where proteins are, there's a little bit of smearing, so we degraded some protein. We treated it pretty harshly. There's some bands here. You can see some fainter bands here. The 40-fold has the same pattern. It just looks like this is a little bit. So around 120 kilodalton, 70, 60, and 13, it looks like are places we could target for isolation. So we are pulling some protein off the surface with that N-octoglucoside, but we have not sorted through yet, is any of these proteins the ones responsible for the conversion. So that's one of my next students' projects. So what kind of things would we like to do with this? Well, after looking at the literature, these little columns here we can put on our HPLC, And these little columns are called luciferase high-trap columns. The material that these columns are made of has a structure similar to that huge NADH. Well, the enzyme binds to those coenzymes because they work together to do the reaction, and there's some reports in literature saying, hey, this has been good for purifying those kinds of enzymes. So I just purchased those recently, and we're going to try that to see if any of the proteins we've pulled off are still fully formed, and if they are, we can isolate them in a single test tube. We'll send them off and sequence it and determine what that protein is, and if it's that class of enzyme. For the biotransformation work, I have students this semester working for me. We need to repeat in triplicate all of the antimicrobial and the yeast work so that we have some degree of certainty of our preliminary results. We'd like to try some other propyryl ketones from the literature, and we have a couple of those picked out, and I have a student that will be working on that. And then this semester, in the last 2 weeks, we've been looking at other vegetables, radish, onion, potato, sweet potato, and apple, trying to make the R isomer, because what we would like to do is compare the R isomer antimicrobial properties to the S isomer antimicrobial properties. So we're working on that. Finally, these are the, one too many. These students here are the three young ladies that really pushed this project forward. I had a couple before them that kind of got it started for us, but they really worked well. So they're all, Marianne here is at TCU working on a PhD. Amy went to work for Dow as soon as she graduated, and Jasmine Moreland is at UH Pharmacy School. I have two new students that are going to be working on that, Nicholas and Jessica Bronstead. Dr. Odanuga is the other biochemist. Dr. Taylor is the fungus lady that helped me. We have a Welch departmental grant, and I had a research grant from the university to help get me started with this. And so with that, I'll be happy to entertain any questions.
Any questions for Dr. Harris?
If you add NADH to your mixture, can you keep using the carrots?
We have tried that once, and it didn't really seem to give us any more yield. So NADH is fairly expensive, and so we haven't tried it again yet. That would be more expensive than just using more carrots.
Right, but I just
So,
didn't know. If
right,
that's
we
why
added,
carrots
we're trying to
started
figure out, like, so we
uses.
added half the molar amount of NADH back to it, and we still didn't get complete conversion. TLC is not the best for a truly quantitative. Qualitatively, it didn't look a lot different for that, so we aren't sure. Yes.
Did you see any difference if you use different portions of the carrot and potato, portions closer to the skin versus?
So for the carrot, there's the internal cortex, there's the phloem part, and then there's the epidermis.
Yeah.
And so originally, not any of the people I showed here, I had a girl that was a botany, just loved botany. So she's like, can I work on this? I'm like, absolutely. And so we did the epidermis. She sliced it under a microscope over in biology to where she took the cortex only, and she took the outer part. All portions made the conversion. The inner cortex, we kept seeing an extra impurity. We never explored what it was. So now we avoid the inner portion. It made the alcohol, but it made something else. We had an extra spot. So we're like, okay, I'm not a biologist, so we'll just stick with the part that makes the single alcohol. So yeah, we've had some people look at that. Yes.
Do you have a number of research projects that you work and collaborate with biologists on?
Really, just this one.
Just this one?
Just this one. So the other biochemist helps me some, and then of course I was trained originally as a chemist, so I'm okay with IR when we did the IR portion of that. And so it's mainly been just me and undergraduates, and then these that come in and help some.
And your Welch grant-supported students.
Yes, they get scholarships for participation. They're required to take 2 for their degree.
Right.
All 3 of the ladies that I showed, they worked with me for about 2.5 years. And the last semester for all of them, they were about the level as beginning graduate students. They were to design their own experiments, and came to me, and we kind of worked out some things to do next.
Were they all chemistry majors?
They were all biochemistry majors. We have 2 degrees. We have chemistry and biochemistry.
Are you looking for students for this summer?
Yes, we're always looking for students for something.
I like carrots.
We have a small master's program too, so if you're interested, we have a master's in natural science that's an interdisciplinary degree. well.
Okay. Well, Dr. Harris will be up here for a few more minutes if you have more questions. Also, we'll be down in the Joyce Room at the cafeteria if you want to join us there to hear more about Stephen F. Austin or carrots or anything else. While you're eating your carrots, you can check on the epidermal and the cortex and all that other stuff and see what kind of stereoisomers you can make out of them. All right, let's give our speaker another... See all of you after spring break.
Let's talk carrots and potatoes and broth and getting hungry.